Hi, this is Alma, and I'm here to give you an introduction to building common coordinate frameworks for spatial transcriptomics data. Now, this idea of a common coordinate framework is a fairly new one, and that means that this field of common coordinate frameworks for spatial transcriptomics data is not quite as established as other topics in sort of like the analysis of spatial transcriptomics data. So my ambition here was actually to try to somehow explain what we actually mean by common coordinate frameworks and try to motivate why we actually need these kind of methods, to then show you one of the methods that I've developed called Eggplant, which is designed to actually build these common coordinate frameworks and discuss a bit how this specific method works, and also try to convince you that it's actually doing the task that it's designed for. So this presentation won't really go through a lot of different methods because frankly there aren't really that many methods out there. So I hope you enjoy it, and let's start by trying to set the scene a bit here. Now, you can imagine a quite common scenario where we have collected tissue samples representing the very same structure but from multiple different sources, such as, for example, different conditions, for example, healthy and disease, different time points, maybe during development, or maybe you just have gathered different replicates to gain more statistical power. Now, very often in our studies or experiments, our objective here is to compare and relate features between our different samples. And I guess examples of projects where this is extremely important or a high priority are these atlas efforts, such as, for example, the Human Cell Atlas. Now, in order to achieve this objective, very often when working with non-spatial data, we tend to employ strategies for batch correction or data integration. We create this low-dimensional embedding based on the gene expression, and then we try to relate and compare observations in the low-dimensional space or the low-dimensional gene expression space. Now, what's really cool when we're working with spatial data is that actually our data, it already possesses an inherent low-dimensional representation, being the spatial domain. And this allows us to extend the idea of integration beyond just the removal of batch effects, but to also be able to relate observations in the physical domain. And this is super cool, but it comes with certain challenges. And I think the biggest one here is sort of like this issue of independent coordinate systems. And what I mean by independent coordinate systems, I think, is actually best explained by an example, so I'm going to give you a very brief one here. So you can imagine that we have collected two different tissue samples from different time points, time point 1 and time point 2. Now, our objective here is to examine how the gene expression at a certain position, x here, changes over time. And as you can obviously see here, the issue is that the coordinates x and y, they don't represent the same position in time point 1 as in time point 2, and that's because we have two independent coordinate systems. The correct approach here would be to compare the expression at coordinates x and y at time point 1 with the expression at coordinates x prime and y prime at time point 2. And one solution to this issue is to transfer the expression to a common coordinate framework where the coordinates x and y actually do represent the same position in all of the different samples. And I just want to take a moment here to elaborate a bit on what I mean with these common coordinate frameworks, or CCFs for short. And a common coordinate framework is something that we transfer information to, meaning that we transfer information from the observed data to this common coordinate framework that allows all of the information to inhabit the same spatial domain. And when I say information here, it could be anything from gene expression to protein signals to even image data. Although our main focus here is of course the gene expression working with spatial transcriptomics data. And now this is very different from doing an alignment, and I've tried to illustrate the alignment procedure here in this image to show you what sets it apart from the transfer of information to this common coordinate framework. And as you can see, even though we've managed to orient our data in the same way and put it in the same scale, the information still inhabits different spatial domains. It's not all in the same common coordinate framework. So going back to this task of transferring information from the observed data to the reference, in our method that I'm about to present, we singularly focus on this transfer process. And what further sets this transfer process apart from the alignment procedure is that we also try to correct for these non-linear distortions, such as, for example, squeezing or elongation when handling the samples, or perhaps the tissues have just grown slightly different, because we all know that there is biological variety between different individuals. Now, before I go into the specific details of the method, I just thought it would be nice to try and explain and elaborate a bit on why we're interested in a method like this. And I've tried to illustrate that in this image here, where we can see how we have observed data in one column and the transfer data in the other column. And the key advantage here of us transferring information from the observed data to the reference is that we now can compare the expression location by location between the different samples, because all of the physical locations now represent the very same position. And this means that differences can be assessed both visually and computationally between the different individuals here. And these different individuals could represent, for example, different ages or perhaps case versus control. So here then comes the million-dollar question: how can we achieve this objective? How can we transfer our information from different types of observed data to a common coordinate framework? And how do we create this common coordinate framework? Well, the method that we designed to do this is called eggplant, which is short for effortless generic GP landmark transfer. And now there is a bit of a fun backstory to this logo that you're seeing here, because this is actually the second iteration. My initial logo was very, very heavily inspired by this eggplant emoji, which I thought was nice, and it has some nice colors as well. And then when I had sort of like a presentation at a course that actually was similar to this one, one of the students afterwards sort of like pinged me on Slack and was like, "Hey, yo, nice presentation and all, and it seems like a nice tool, but just so you know, FYI, maybe your logo could sort of like have certain unwanted associations." And yeah, that sort of like hit me then, and I realized I had to redesign this logo, and this is now the final product, which I hope has no such unwanted associations. So with that being said, let us now have a look at how this method works. And you can more or less summarize eggplant in 5 steps, where we start off by constructing or choosing a reference that we want to transfer information to, and then we select the samples that we want to transfer information from to this specific reference. Next, we chart the landmarks, meaning that we annotate common landmarks found in both the observed data and in the reference. We then select a feature of interest, for example, the expression of our favorite gene that we want to transfer from the observed data to the reference. Then we learn a transfer function relating landmark distances to the gene expression of this favorite gene. And finally, we transfer the information from the observed data to the reference by applying this learned transfer function from step 4 to each location within the reference. And now there are 2 parts here that I would just like to spend a little bit more time on, because it might not be super clear what I mean from this very overviewish description that I just gave. And those are the landmarks and this specific transfer function. So let us start with the landmarks then. And I think a fairly legit question here is sort of like, what is a landmark? And here we define landmarks as structures, signals, or any feature that can easily be identified across the different samples. And this definition is largely based on what is presented in this really nice perspective in Cell, called Toward a Common Coordinate Framework for the Human Body by Root et al. And if you're interested in this sort of like concept of common coordinate frameworks and how it can be applied in, for example, the Human Cell Atlas, even for single cell data, I think this is a really nice publication that you can check out. Now, a key feature here, or a key point, is that a landmark should always represent the same approximate position. And landmarks can, for example, be based on the morphology if you have an image associated with your data. It could be based on the gene expression itself if that helps you to pinpoint certain locations with your data, within your data. Or it could be based on protein signals if you have, for example, a complementary stain that you use on your spatial transcriptomics data. Anything that allows you to identify a certain region or position can be used as a landmark. Now, what's really important here is that eggplant only focuses on the transfer process, meaning it's completely agnostic to the annotation method. And so far, all of the examples and results that you will be seeing here have been manually annotated by a non-expert, aka me, which is actually really nice, because if I can get this method to work with my crappy ass annotations, anyone with the slightest clue about the biology of the samples that they're analyzing should be able to sort of like get this to work without any issues. But we're also working on sort of like developing these automatic annotation methods, but that's a slightly different project. Now, just to give you an example of what could constitute a good landmark, if we look at this sort of like hemisphere here of the mouse brain, this tip of the dentate gyrus would, in my opinion, be a really nice landmark, because it's very easy to spot and identify just based on their morphology, and assuming that you're at the same depth within the mouse brain, it corresponds to the same approximate different position within all of the different samples. Okay, so those are the landmarks. Let us now have a look at this transfer function, which is actually the core part of this whole method. So we learn this transfer function by using the observed data. And what we do here is that for each location within our observed data, we measure the distance to all of the different landmarks to create this distance matrix X. We then assume that the gene expression Y is related to the landmark distances via a transfer function F. And we also assume that this function F follows a Gaussian process distribution. And I just want to clarify here that this is not a normal distribution or a Gaussian distribution. This is a Gaussian process distribution, which is sort of like a distribution over functions. And with this assumption, we can then use Gaussian process regression to determine the character of this transfer function F. Now, once we've learned this transfer function from the observed data, we can move on to the reference to actually try to transfer the information from the observed data to the reference, and this would constitute step 5 in the workflow that I presented previously. And for the reference, in order to transfer the information, we do something very similar to what we did for the observed data. We create this new distance matrix X bar, where we measure the distance from each location within the reference to each of the landmarks within the reference. And we then transfer the information from the observed data to this reference by applying the transfer function that we learned to this X bar, allowing us to predict the gene expression at each location within the reference. And here you can sort of like see the whole flow of information from how we learn the transfer function in the observed data to how we predict or infer the gene expression in the reference. So I've now been speaking quite a lot about why we're interested in a method like this and how our method works. So I think it's time for me to also show you some results to try to actually convince you that the method actually does work. And with that, I want to demonstrate how Eggplant manages to do 3 things. And that is how it handles inter-individual variance, how it preserves intricate spatial patterns once it transfers information from the observed data to the reference, and how it enables informative downstream analyses. And we'll start off by looking at how the method handles this inter-individual variance. And to demonstrate this, we're going to be looking at a dataset of human developmental heart. And here you can see there are 7 different sections, and maybe it's quite obvious, but as you can see here, there are 4 sections from individual 1, and there are 3 sections from a different individual, which we name individual 2 here. And these small stars that you see in different colors, those represent the landmarks that we have annotated in this specific dataset. And below here you can see the reference that we want to transfer information to from this observed data. And in this case, the information that we want to transfer is gene expression. And now in this reference, you see how there are differently colored regions, and for now, you can just ignore them, but I will come back to them later on. And here you can see what the original observed data looks like before we did any form of transfer to this reference. And I must admit, when I initially looked at this data set, I was like, oh man, this will never work, because if you look at the gene expression and just the spatial structure in individual 1 and compare it with those sort of like sections from individual 2, there are some quite stark or quite distinct differences between these 2 different individuals and the different sections from them. So I was quite pessimistic, but since Eggplant is a very sort of like fast method to run and use, we applied it to this dataset, and the results were actually really encouraging. And you can see them here, and what's really nice is how once we cast the information into the same reference here, the gene expression looks very similar between individual 1 and individual 2, and there's a really good agreement between the samples from the different individuals. And now, even though this is nice to look at, it's quite a lot of information looking at 7 different sections and 4 different genes. So what's really nice now is that since all of this information or gene expression now lives or inhabits the same spatial domain, we can actually summarize all of this information in a single representation, which we tend to call a composite representation. And this is sort of like an average of the gene expression from all of the different samples represented in one single image. Something else that's pretty cool that we can do is to compute this location-wise correlation across the 7 different sections. And just as we expected, we saw a high correlation when looking at the gene expression in sections from the same individual. But what was really impressive and encouraging here was that when we looked at the gene expression in sections from different individuals, we also saw a really high correlation. Now finally, we're going to come back to this reference with the differently annotated regions here. And I just want to make it clear that we didn't do any annotation in the observed data, we only annotated these regions within the reference. But since we've sort of like transferred all of the information to this reference, we can see how the gene expression of different genes varies between these different regions, allowing us to make sort of like a regional enrichment analysis. And this, of course, allows us to assess the up or down regulations of certain genes in different regions. And this allowed us to identify this left-right asymmetry between the different atria in the heart here when looking at the COL2A1 gene. And I think that's a pretty cool example of what you can do once you've transferred information to this common reference. The next thing I like to demonstrate is how eggplant preserves these intricate spatial patterns when we transfer information from the observed data to the reference. And here we're going to be using a different dataset, and this is a sagittal section of the mouse brain, and it's public data from 10x Genomics. And what we have here are two different sections, and these are replicates. Essentially they are consecutive sections more or less. And we are looking at three different genes here: MBP, CALB2 and DNER, and we want to transfer this information contained in the observed data to this reference that you see to the right there. And this is the result of transferring the information contained within this observed data to the reference. To the left you have the observed gene expression, and to the right you have the transferred gene expression. And to me there are two key observations that I would like to discuss a bit further here. And the first one is how the complexity of the expression patterns is preserved once we've transferred information to the reference. You can see these intricate spatial patterns that were present in the observed data, and they haven't been distorted or anything like that. The second observation is that we see a really good agreement between the different replicates, and this is really comforting because we're working with sequential sections here, and we would expect the transferred information to have more or less identical spatial structures once we cast it into this same reference. Now the last and perhaps most exciting part here that I want to demonstrate is how eggplant enables informative downstream analyses. And for that we're going to be using yet another data set, this time from a publication published by Bussi et al. And in this publication, or in this study, the authors injected mice with partially nothing as a control, with a saline solution as a sham, and a heme compound, which they wanted to assess the sort of like activity of in these mice. And they did a lot of really cool things here, but what's of interest to us is that they produced some really nice Visium data here. And we can think of this dataset as a perturbation dataset, where they have perturbed the normal state by injecting this saline or hem solution compared to the control where nothing has been injected. And I won't really go into any details of the biology here. If you're interested in that, have a look at the publication. What's of interest to us is that we have these three different conditions that we could look at and compare different features between. So here's a similar image to what I showed you before with the sagittal mouse brain, where we have the three different sections that constitute the control, the heme and the sham sample. And in the middle part you can see the observed gene expression of three different genes that I highlight in the publication. And these are SERPINA3N, CD63 and TIMP1. And to the right you have the reference that we want to transfer all of this information to. And this is really an ideal dataset for eggplant or any method that tries to create this CCF and transfer information to it. Because here we have three sections that represent the same region or part of the mouse brain, but the spatial structure of these three different sections are quite different, and you couldn't really just align these samples. You really do need to transfer the information to a common reference in order to be able to make spatial comparisons. And that sort of transfer information is what you can see here in the results. So again, to the left we have the observed gene expression of these three different genes for three different conditions. And in the right part you have the transferred gene expression for the same three genes in the same three different conditions. But now you can see how all of the information inhabits the same spatial domain. And I'm now going to introduce to you a kind of analysis that we call spatial differential expression analysis. And the way I'm going to do this is to go by an example using this SERPINA3N gene here. There's nothing super special about this gene, but I think it constitutes a good example. So what you see here in these three images is how we have compared the gene expression on a location-wise basis between the different conditions. So for example, looking at the middle part here, we compare the heme condition with the sham condition. And wherever you have a red signal, there's a higher expression of this SERPINA3N gene in the heme condition compared to the sham condition. and vice versa where you have a blue signal. And the gray regions here indicate non-differentially expressed regions or positions within these samples. And the way that we compute these sort of like signals here or generate these images is extremely simple. Since all of the information inhabits the same spatial domain, we can simply subtract the gene expression of one condition from the gene expression of a different condition. And we do this on a location by location wise basis. And what I really want to emphasize here, that this kind of analysis, this spatial differential expression analysis, is only possible when the information inhabits the same reference or common coordinate framework. So I hope that I've now managed to actually convince you that Eggplant successfully can accomplish all of these three different objectives. And if not, I'm really happy that the reviewers for this paper have already been assigned. So to summarize this presentation and the idea of common coordinate frameworks, we can say that common coordinate frameworks, or CCFs, allows us to relate spatial observations across different samples, for example, different time points and conditions. And our method, Eggplant, transfers information from the observed data to a reference or common coordinate framework. It uses landmark annotations and Gaussian process regression to do this. And transfer of information to a common coordinate framework enables a lot of really impressive and relevant downstream analyses, such as, for example, spatial differential expression analysis, or SDA for short, which I exemplified in the Perturbation dataset. We can also do this aggregation of information, like I showed with the developmental heart dataset, where we create these composite representations of our data. And finally, having a common coordinate framework and casting information into this same reference is imperative to conduct true spatiotemporal modeling, which was actually one of the main reasons that I first got interested in this specific project. Now, if you're interested in more information about this specific tool, Eggplant, you can check out the bioRxiv publication. It's called A Landmark-Based Common Coordinate Framework for Spatial Transcriptomics Data. There's a GitHub page with quite a lot of instructions and information about how you can install this software and also how you run it, and there are some examples of the analysis, for example, all of the analyses that I showed you here today. And there's also a Read the Docs page if you want to look at the documentation and how to use this specific package in your own analysis. And of course, I want to give some acknowledgements here, and that is to Shanetta and Xiaofei and Eric, who produced the developmental heart data, to Paulo, who you all know by now, who did some really nice beta testing of the software and also contributed with some really nice discussions, and of course to my previous supervisor, Joachim Lundeberg. I also want to give a special shout out to Giovanni Palla, one of the main developers for the SquidPy package in Python. And he gave some really excellent feedback on this software that really allowed me to take this software to the next level and enhance its performance. And that's about it for this presentation and introduction to common coordinate frameworks. I hope you found it interesting and that you again learned something. I really appreciate you listening, and if you have any questions, you could either contact me by mail or maybe just send me a DM on Twitter. As before, you can find me on Twitter and also on my website if you're interested in to see what I'm up to or so like what I'm doing at the moment. Thank you very much and enjoy the rest of the course.